Hello, my name is Samuel Pineda, and I'm an incoming biomedical engineering freshman at the University of California, Irvine, and I want to quickly talk about recent studies involving DRT3 and its implications on the central dogma theory as well as DNA writing technologies. Now, the central dogma theory is a framework which describes how genetic information is created and flows within all living organisms. DNA can create RNA via transcription, and RNA can create proteins via translation. It also describes how DNA can only be created by either replication or reverse transcriptase using RNA as a template. Now, for decades, we thought these were the only ways to create DNA, until very recently when Stanford scientists published earlier this year findings that show possibly a new way to create protein involving DRT3. Now, DRT3 stands for Defense Associated Reverse Transcriptase 3, and is part of a family of newly discovered bacterial immune systems. Now, this protein in particular is very interesting. It can produce and synthesize DNA using itself as a template. Now, how does it do that? This DNA has amino acids that mimic the nucleic acids of RNA strand templates. And by mimicking these nucleic acids, it can utilize reverse transcriptase to create single-stranded DNA. Now, while it seems that these proteins can only synthesize one specific strand of DNA, and the purpose of these DNA strands are not currently really understood. Scientists theorize that these DNA strands act as some sort of sponge and latch onto phages within the bacteria to either hinder them or to act as some sort of marker for immune elements within the bacteria. Either way, this is a huge discovery. While it does not necessarily upend the central dogma theory, it does demonstrate that there could be other ways to create DNA outside of what we previously knew. There could be ways that DNA is produced in nature that we do not know of. Furthermore, it has further implications in biotechnology. Chinese researchers from universities in Shanghai highlighted how similar this is to CRISPR technology. CRISPR involves splitting DNA and inserting customized strands. This technology was actually derived from bacteria immune systems. However, there are two main differences between this and DRT3. CRISPR technology involves splitting and editing existing DNA and utilizes nucleic acid bases, while DRT3 creates completely new strands using amino acid bases. So because of this, there are many possible use cases in the future. One main one is that we could replicate CRISPR technology, but instead of using nucleic acid bases, we could possibly use amino acid bases. Another interesting use is that because DRT3 creates completely new strands, we could possibly synthesize DNA materials such as DNA hydrogels. Now, while we do need a lot more research, this is an extreme breakthrough for the scientific community. And these are my references. Thank you for your time.